Hello, welcome to Chapter 25, Metabolism and Nutrition. So we're going to go through what happens to all of these different nutrients after they have been absorbed into our system. What do we do with all of these different things? How do we make energy? We're going to look into this today. So your metabolism is known as the sum of all chemical reactions that are occurring in your body. We can have catabolism, where we break things down. Some can be exergonic, some of these catabolic reactions, which means they're going to produce more energy. They produce more energy than they're going to consume in the process of breaking down these different chemicals. This involves glycolysis, the Krebs cycle, and the electron transport chain. We're going to talk about all of these today and go into what we're breaking down, how we break it down, what our end products are. Anabolism. This is where we build something. We take something that is in its simple form and we make it into something more complex. This is how we make proteins. We build our different fatty acids. These are endergonic. It takes more energy to build something in our system than it does to break something down. We have to consume energy to build something, and we don't get a whole lot in return for that. So our metabolism is actually a balancing act between building stuff, anabolism, and breaking stuff down, catabolism. ATP, or adenosine triphosphate, is the molecule that we use for energy. We have our adenosine, and then we have 3 phosphates. And these phosphates are all bonded together here. Adenosine triphosphate, we use catabolic reactions, and then to break it apart, and we will use anabolic reactions to put it back together again to make more energy. So these different reactions that we do throughout our entire body actually depend on the amounts of energy and passing this energy from one molecule to another. ATP is always thought of as that energy currency in our body. We use this ATP for everything that occurs within our body. We talked about it with muscle contractions in the skeletal system. We've mentioned it in several other areas. Now we're going to talk about it again. So cells usually have about 1 billion molecules of ATP. Those billion molecules of ATP only lasts for less than a minute. So we have that adenosine. There's technically a ribose molecule in there, and then there's those 3 phosphate groups. These 3 phosphate groups, we can break off phosphates. So these 3 phosphates, adenosine, phosphate, 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 we break one of those bonds. That's what'll give us this ATP that equals adenosine diphosphate plus phosphate plus that energy that we gained from breaking that bond. Some of this energy will be used for anabolic reactions where we build things up, like building glycogen. Glycogen is those chain of glucose molecules that we store. So glycogen we use this ATP through anabolic reactions to make. We catabolize when we take this ADP plus that phosphate to remake ATP. So the body has a continuous need for energy. We always need energy. It is there to help us sustain our life. It keeps us going. This energy is required for every single function in our body. So we have these different molecules. We can break them down. We can build them up. ATP is the same way. We break it down to adenosine diphosphate plus phosphate. We can put it back together again. So these cycle kind of goes in a circle as we break something down or build something up, and the energy that is required to do all of those different reactions. So an oxidation reaction is when we remove electrons, and it'll give us a decrease in the potential energy of that atom or that molecule. Most oxidation reactions in our body are known as dehydrogenation reactions. So we take lactic acid, we put it through an oxidation reaction, and we get pyruvic acid. When our muscles are exercising, if we don't use the pyruvic acid that comes out of glycolysis, our body converts it to lactic acid and releases it out into the bloodstream. Our liver and our heart can take this lactic acid. The heart can take the lactic acid and create ATP directly. The liver will take the lactic acid and turn it back into pyruvic acid. These reduction, this was one I was just talking about. We add electrons in a reduction reaction. So we increase that potential energy. So that pyruvic acid, it's what's left. It's the end product in glycolysis. If we don't use it to make energy with, it turns into that lactic acid, which then gets released out into our bloodstream. So when something gets oxidized, these hydrogen atoms, they get transferred by some coenzymes. So they could be put onto nicotinamide adenine dinucleotide, or NAD, or flavin adenine dinucleotide, or FAD. So this NAD is going to become NADH. This FAD becomes FADH2 because there's two hydrogens. This is what happens when we are going through the different steps in the process to create ATP within our cells. So when we take this NAD, reduce it to NADH plus H plus that hydrogen, that NAD plus gets this hydrogen or hydride ion, and that neutralizes that NAD plus. Then that hydrogen, that positive ion, gets released out into that solution. When NADH gets oxidized, it's going to turn into that NAD plus. So we get rid of that hydride. So we have one less oxygen, and that's what, or hydrogen, and that's what makes NAD plus, that positive. Because if we look at this hydride, this hydrogen here is negative. So we're taking this one positive, this plus sign up here on that NAD is telling us that this NAD has a +1 charge. We take this hydrogen with that negative charge, a +1 and a -1, evens or neutralizes it out. FAD, flavin, is reduced to FADH2. It's going to gain a hydrogen atom, and it gains a hydride atom. That hydrogen is a plus, that hydride is that negative. So that plus and that minus, +1 -1 is 0, is a neutral charge in this case. So that's what gives us the FADH2 with no charge. FADH2 can be oxidized into FAD. It's going to lose that hydrogen and that hydride. They're always coupled together. So when something is oxidized, something else is being reduced. This is known as this oxidation reduction reaction, or these redox reactions. Lactic acid gets oxidized to pyruvic acid. 2 hydrogen atoms get removed from this lactic acid when it gets converted to pyruvic acid, and they're used to reduce NAD+. So here's this oxidation reduction. We take this lactic acid, we reduce it down to pyruvate. We're taking off hydrogen and a hydride off that lactic acid. So now that gets put onto this NAD+, which will give us NADH plus hydrogen. Oxidation tends to be exergonic, so we release energy. So when we oxidize glucose, C6H12O6, the energy in glucose gets removed, but it takes several steps to get there. Some energy is made in the process. Some energy is used in the process. So these compounds with many hydrogen atoms, like glucose that has those 12 hydrogen atoms, they have more chemical potential energy than oxidized compounds. So some of this energy that is released, we capture when ATP is formed. We add that phosphate group from ADP to get ATP. This is this phosphorylation, which is that addition of a phosphate group. So there's 3 ways this phosphorylation can generate ATP. We have substrate level, oxidative phosphorylation, and photophosphorylation. Substrate level phosphorylation is going to generate ATP by taking that phosphate group and attaching it directly to ADP. This is happening in the cytosol. This is happening during glycolysis. Oxidative phosphorylation removes electrons. When we remove electrons, series of electrons, they get attached to these electron acceptors. This is during this electron transport chain, which is part of the step in this energy or ATP generation of many ATP generation. This is done in the presence of oxygen. This is part of the Krebs cycle, and that, well, the electron transport chain specifically is in that mitochondrial membrane. Then that photophosphorylation is in plants and bacteria only where they can absorb light. Our body utilizes this glucose for making ATP. It's our body's preferred way. We have all the different systems and steps in place to take ATP and generate glucose and generate lots of ATP. So ATP production, this helps the cells that are making that ATP because we can take glucose and oxidize it into ATP. We can even do that when ATP is not needed yet. So glucose can enter one of several metabolic pathways. We have amino acid synthesis. So we can use glucose to form amino acids to make proteins. Glycogen synthesis. Livers and skeletal muscles are the only place that go through glycogenesis. We break down this word. Glyco is for glycogen. Then genesis means we are making something. So glycogenesis, we are making glycogen. We are stringing hundreds of glucose molecules together. The liver can store about 125 grams of glycogen, while the skeletal muscles can store about 375 grams. So we can keep a pretty large supply of glucose ready. Here's the other fun part. Glucose can get converted to triglycerides, to fat. So when our glycogen stores are completely filled, glucose can transform to glycerol and fatty acids for lipogenesis. Lipo for lipids. Lipids are fats. Genesis means to build something. Lipogenesis, we're building lipids or fats. We do this to make triglycerides. We store those triglycerides in those adipose cells. Those adipose cells have pretty much unlimited storage. They can continue to expand and expand and expand. Before we can use glucose, our cells need a way to get glucose inside of the cell. We absorb glucose in our digestive canal through that secondary transport. Most of the time we use these GLUT molecules and we utilize facilitated diffusion to get glucose across the cell's membrane so we can start the process of making energy. So glucose catabolism, we first start with the oxidation of glucose. So we're forming ATP with cellular respiration, which should have been mentioned in the skeletal muscle chapter. We start with glycolysis, and this is a several-step process where we take one glucose molecule and we break it down and get 2 molecules of pyruvic acid as our final products. We also get 2 molecules of ATP. So we get some ATP. We don't get very much from glycolysis. It does not require oxygen. So this can be the first step in aerobic respiration, or it is the only step in anaerobic respiration. We also get 2 NADH plus H out of glycolysis. The next step is the formation of acetyl-CoA. This is where we take that pyruvic acid and we get it ready to go into the Krebs cycle, also known as the citric acid cycle. And for this pyruvic acid to enter the citric acid cycle or the Krebs cycle, we need to make acetyl-CoA, coenzyme A, or acetyl-CoA. This is what we use to put into that Krebs cycle as the next step. This is where we start the aerobic respiration, where we start using oxygen. At this point, after we've put these two acetyl-CoAs, because we have two pyruvates, there's two molecules of pyruvate. So each pyruvate is going to make one acetyl-CoA. So each of these acetyl-CoAs will go into the Krebs cycle. We do make another NADH molecule in that formation of acetyl-CoA. So now at the Krebs cycle, we take that acetyl-CoA or coenzyme A, we put each of those acetyl-CoAs through that Krebs cycle. So we take 2 turns through that Krebs cycle. Krebs cycle is a circular chemical reaction that is happening. So each acetyl-CoA is 1 turn. The second one gives us a second turn. Here we get carbon dioxide, some ATP, some NADH, and we get FADH. The next step is the electron transport chain. This is the last step in cellular respiration or aerobic respiration. Now we are taking all of these different NADH molecules that we've made. We have 2, 3, 4 NADHs. Then we get an FADH. We are putting these into the electron transport chain. During this process of sending these NADH plus H and that FADH2, we are taking these electrons off of that NADH plus hydrogen and that FADH2, and we use those electrons to generate ATP. So here we're able to generate very large amounts of ATP from that original glucose molecule. But it takes glycolysis, the formation of acetyl-CoA, the Krebs cycle, and the electron transport chain for all of this to happen. So that aerobic respiration requires oxygen. Glycolysis, we can throw oxygen into. Krebs cycle and the electron transport chain need oxygen. Anaerobic, like weightlifting, is without oxygen. We have glycolysis without oxygen, forms lactic acid. So let's go through these different reactions here. So we start with glycolysis. Glycolysis takes that glucose molecule, and we are going to split this 6-carbon molecule into 2 3-carbon molecules, which gives us that pyruvic acid. It takes 2 ATP molecules to split that 1 glucose molecule, but it provides 4 ATP. This is how we net 2 ATP, because we get 4, but we use 2. So after we subtract out what we use to get that, we end up with 2 ATP molecules at the end. Glycolysis is a series of steps that go down that will give us those 2 pyruvic acids and then those 2 ATP. Pyruvic acid, if there is oxygen available, we will take those pyruvic acid and put it into the lactic or the citric acid cycle. But first we have to turn it into that acetyl coenzyme A that is kind of the middle step between glycolysis and the Krebs cycle. This is if oxygen is available. If oxygen is not available, that pyruvic acid gets turned into lactic acid right away. It diffuses into the bloodstream, and the liver can turn it back into pyruvic acid to be used to make that acetyl-CoA to go through the citric acid cycle. So here's glycolysis. It's a series of several different steps. Knowing that we take one glucose molecule, we get these two pyruvate molecules, and we're going to get 2 ATP in the process. We also get these 2 NADH molecules. These 2 NADH molecules will come in handy at the electron transport chain. So here is technically all of the different steps of glycolysis. I am not going to ask you to memorize this. If you take biochemistry classes as part of whatever program you are trying to get into, you will have to memorize glycolysis. Here, just knowing that we take that glucose molecule, we split it, get 2 pyruvate, 2 ATP, and 2 NADH plus hydrogens. That's all you need to know. Now we take those pyruvic acid. We have oxygen available in our cells for this example. Now we're going to take that pyruvic acid and make this acetyl coenzyme A. We take a coenzyme from cellular respiration. Between glycolysis and the Krebs cycle is where this pyruvic acid gets converted into this acetyl group by removing carbon dioxide. This is known as decarboxylation. So each pyruvic acid loses 2 hydrogens. This is where we form NADH plus hydrogen. And then we form 2 acetyl coenzyme A's, or acetyl-CoA. We form 2, 1 for each pyruvic acid. So during this step-by-step process, we've got our pyruvic acids. This pyruvic acid goes inside of the mitochondria. Glycolysis happens in the cytosol. Once we get those pyruvic acids, it goes into the mitochondria. It gets converted into that acetyl coenzyme A by adding that acetyl group, getting some NADH plus hydrogens. We add that coenzyme A on to that acetyl group that pyruvic acid was converted into. And now we have 2 acetyl-CoAs that we can put into the Krebs cycle. That Krebs cycle, or the citric acid cycle as it's also known as, will only let these acetyl-CoA molecules in. So this is another series of reactions that happens. This one's more circular. It goes in a nice circle. We produce 6 NADH, 6 hydrogens, 2 FADH, and 2 more ATP molecules for each acetyl-CoA. So that's quite a bit. So this Krebs cycle, it's a series of reactions that goes through a nice big circle. It generates all of these different NADHs. It generates this FADH. And these are all working to create these 2 ATP that we get out, and then these 6 NADH and this FADH. So here's all the step-by-step reactions. Again, if you take any biochemistry class, you're going to have to memorize every single step of what is happening, what molecules are going in, what's coming out. Here, just knowing that we take pyruvic acid, We convert it to acetyl-CoA. We put it into the Krebs cycle. We get NADH plus, NADH plus, hydrogen plus, FADH, NADH plus. We get several of these. We get 2 ATP in the process. And then we're going to go to the electron transport chain. We take all these NADH plus hydrogens and this FADH2. From here, all of these NADH plus hydrogens and this FADH2 go through a series of electron carriers. These series of electron carriers is going to take electrons off of these NADH plus hydrogen and this FADH and use it to add these phosphates onto these ADP to get ATP. So chemosis is this mechanism of ATP generation where we take through these chemical reactions pumping of hydrogen ions that are going through. These hydrogen ions are these electrons that are going through this electron transport chain and adding phosphates on. We also have proton pumps that are part of this electron transport chain. So this is going to pass any of the energy from this NADH plus hydrogen, and they're going to pump these hydrogen molecules from or through these mitochondrial membranes. If there's with a high concentration of all of these hydrogens that are coming from this glucose molecule that we have thrown through all of these different steps that have been broken down, this ATP synthesis occurs when these hydrogen ions kind of flow back into the mitochondrial matrix. So here's what is happening here. So we take NADH plus hydrogen, we put it through this electron transport chain. We take these hydrogen ions that are coming off, which are also the electrons that are coming off, with this proton pump pumping all these hydrogens through. The energy from breaking these hydrogens off are attached to this ADP plus phosphate to make ATP with the enzyme ATP synthase. So this is more the whole process. Again, if you take a biochemistry class, you're going to have to memorize every aspect of this. Here, just knowing that we take that NADH plus hydrogen and that FADH2, we put those in and we can generate ATP. That's as far as we need to know. So we start with this one glucose molecule. It takes 6 hydrogen atoms. We can make 30 to 32. We take 30 to 32 ADPs and 30 to 32 phosphates. Our end product, 6 carbon dioxides, 6 water molecules, and 30 to 32 ATP for 1 glucose molecule. Glucose anabolism. So if we have more glucose in our body than what is needed, we don't need it for ATP production right now because we're laying on the couch doing nothing. We are going to form hydrogen. And through this process, we use the insulin that came from the pancreas, and that gets these hepatocytes and these skeletal muscles to go through glycogenesis. Again, glycogen, genesis, building, we're building glycogen. And we just keep adding more glycogen molecules on or glucose molecules onto this chain of glycogen until the liver and those skeletal muscles have reached their limit. At this point, what we will do is start converting glucose to amino acids, converting glucose to fats. So what happens to this glycogen that we now made when we need glucose? Our blood glucose levels get low. Well, glycogenolysis. This is when we break down the glucose and release it into the bloodstream. It requires energy. So each of these glycogen molecules, or glycogen, these chains of glucose, we're breaking off these individual glucose molecules. Glyco for glycogen. Lysis means we're breaking it apart. So I'm going through giving these root words because knowing what these words mean, like glyco for glycogen or lysis, breaking down, genesis, building. If you understand that much, it'll help you on the exam. It'll help you in further classes, just being able to break down the words to know those individual portions. You can figure out what pretty much any word means. That's the nice thing with, say, medical terminology, is once you know what these different parts of these words mean, when everything is put together, you can figure out what something is supposed to mean, even if you've never seen it before. So back to this glycogenolysis. We split off glucose molecules using phosphorylation. Well, what about carbohydrate loading? If you've ever talked to any longer distance runners, like marathon runners or beyond, when they're going to do a long run the next day, or then over the next like 2 to 3 days before this longer run, like marathon runners do this for 2 to 3 days leading up to a race. They are trying to increase the amount of glycogen stores that they have in the liver and these skeletal muscles. So what they will do is, over these 3 days before, they will eat very large amounts of complex carbohydrates like pastas, potatoes, for these 3 days. This is known as that carbohydrate loading. So it kind of helps to maximize the amount of glycogen that they have readily available in their muscles to be broken down for energy. So any athletic event that lasts more than an hour, people may carbohydrate load for, because it helps to increase their endurance so they can go for longer amounts of time without needing to stop to eat or take in any kind of calories. So the longer they increase the endurance, they increase the glycogenolysis, and it results in more glucose that is readily available to be used for energy. So what happens when we run low on glycogen? Well, one bonus is we can start breaking down triglycerides for energy. We're breaking down fats for energy, that's a bonus. But once we get to the part where we start breaking down proteins, that usually means we're breaking down different organs. We're breaking down different parts of the body that we need, that we use. So we do catabolize or break down these triglycerides and proteins, but these triglycerides and proteins, it doesn't happen unless you are starving. If you eat very few carbohydrates, or if you have some sort of endocrine disorder. So what happens, this glycerol, part of the triglycerides, lactic acid, And amino acids can be converted to glucose in the liver through what is known as gluconeogenesis. Glucose, new, genesis, forming new glucose. And as this happens, we can take that pyruvic acid, we add glycerol, we can add certain amino acids, or we can add lactic acid and convert it back into glucose to make energy. So lipids, those different fats. Most of our lipids are nonpolar, hydrophobic molecules. They don't like water. Fat, oils, they do not dissolve in water, so we have to make them water soluble. How we make lipids water soluble is attaching them to proteins or making them lipoproteins. These lipoproteins are transport vesicles that transports fats in the bloodstream to somewhere else in the body. We have some different types of lipoproteins that do this. We have very low density lipoproteins, VLDLs. They're made by hepatocytes, and they transport these triglycerides to adipose cells, those adipocytes. So once they deposit their triglycerides in these adipose cells, they become LDLs. We have these low density lipoproteins, LDLs. They carry most of the cholesterol in our blood. They're going to deliver this cholesterol, fats, to different cells for repair purposes. They can make steroid hormones. The liver's going to make bile salts. So once the cell has enough cholesterol for activity, it sends out these negative feedback mechanisms that are trying to inhibit more of these low density lipoproteins from being made. If we have too much of these low density lipoproteins, they can start to deposit cholesterol around smooth muscle, around arteries. So we're starting to deposit fat in different areas of the body, just trying to get rid of it somewhere. This is where we can form fatty plaques in our blood vessels that can cause coronary artery disease. Since these LDLs will deposit bad cholesterol anywhere they feel like in the body, they are known as the bad cholesterol because they don't have a cell to take it to. They will just take it wherever they feel like and dump it. High density lipoproteins, HDLs, they remove excess cholesterol from our cells. They transport it to the liver for elimination. So they prevent that accumulation of cholesterol in the blood. HDLs are considered the good cholesterol. You want higher levels of HDLs and lower levels of LDLs. Since these HDLs are preventing this cholesterol, these fats, from being deposited anywhere in the body, wherever it sees fit. So we have some sources of cholesterol in our own body, like the hepatocytes in the liver. They can make cholesterol. We also get cholesterol from eggs, dairy, if you eat any organ meats, beef, pork, processed lunch meats. fatty foods that don't have cholesterol can actually still increase your blood cholesterol levels. Because these dietary fats, they actually stimulate the reabsorption of cholesterol containing bile back into the blood. So when saturated fats get broken down, hepatocytes use some of these to make cholesterol. A blood test, known as a lipid profile or a lipid panel, that's the other name for it. They're going to measure total cholesterol, HDL level, usually LDL level, and triglycerides. Usually that LDL is calculated based off of the total cholesterol found in your blood minus the triglycerides divided by 5. The total cholesterol in an adult should be under 200. So as the amount of cholesterol in your blood increases, your risk of coronary artery disease also rises, gets much higher. There's different ways to help to lower those cholesterol levels. Your diet, eating foods that contain less fats and less cholesterol. Exercise, always. Exercise is recommended for most anything. And medications. Aerobic exercise or near aerobic exercises actually helps to raise your HDL levels, those good cholesterols. We work to try to reduce the total amount of fat in your system through exercise. There are different medications that can block some of these enzymes used to make cholesterol. So it helps your body try to get rid of these excess fats. But our lipids can also be oxidized to make ATP. We get a lot more ATP from lipids than we do technically from glucose. It just consumes a lot more ATP and it takes longer for it to happen. So if your body doesn't have that need to use lipids for ATP, they get stored around the body. If it is needed, what will happen is we will break down those lipid molecules to make ATP. There are 2 essential fatty acids that our body cannot make. These are fatty acids that we actually have to consume. These 2 essential fatty acids are linoleic acid and linolenic acid. These are 2 essential fatty acids. Our system cannot make these. We have to consume them. They are found in vegetable oils and they're found in leafy vegetables. This is the part of the importance of getting a nice balanced diet, because this is how we get some of these different essential fatty acids for our system. So triglyceride storage. So our adipose tissue, its function is to remove triglycerides and store them until they are needed for ATP production. Triglycerides get stored in those adipose tissues, and it makes up about 98% of all of our energy reserves. Triglycerides are stored and they're more readily available than glycogen because they are hydrophobic. They don't have any kind of osmotic pressure that's put on these adipose cells. Other functions of our adipose cells is to insulate. They do protect various body parts and organs. And this is why everyone needs to have a certain minimal amount of adipose tissue in their body, because we need to protect our organs. We protect our body. Those subcutaneous adipocytes, those subcutaneous fat cells, they have about 50% of those triglycerides that we've got stored. They're constantly being broken down and resynthesized, remade. So they're continually broken down, resynthesized, broken down, resynthesized. This is happening all the time. So during lipolysis, lipo for lipids, lysis, we're breaking them down. This is when we use those triglycerides for ATP. And first they have to be split into glycerol and fatty acids. This is part of this lipolysis. We catabolize with lipases or enzymes that are used to break down these fatty acids. Epinephrine and norepinephrine actually enhance lipolysis. So during exercise, when we release that epinephrine and norepinephrine during exercise, this is what causes us to burn fat. Cortisol, thyroid hormone, insulin-like growth factor, they also help to stimulate lipolysis. Insulin inhibits, because when we have insulin, we're trying to take glucose up into the liver and our skeletal muscles to create glycogen to store. So that glycerol and those fatty acids that we are left with after that lipolysis, they are broken down themselves. So glycerol gets converted into glyceraldehyde 3-phosphate, which gets converted to glucose. So if our ATP levels are low, this glyceride 3-phosphate enters this pathway to get to pyruvic acid. Now these fatty acids go through what is known as beta oxidation. It's a series of reactions that converts fatty acids into that acetyl-CoA. We skip over glycolysis with these fatty acids, and they can enter into that Krebs cycle, then down to that electron transport chain. So here's all these different steps that glucose can take, that these fatty acids can take. These fatty acids go through beta oxidation and go directly to acetyl-CoA. They can also go back to being fatty acids and being stored if we don't need them. Lipogenesis. Lipo for lipids, genesis, we're building. We're building lipids. So our liver and our fat cells, those adipose cells, they can make lipids from glucose, amino acids, through this lipogenesis. Insulin causes us to build fat. So it happens when somebody consumes more calories than they need to satisfy their ATP needs. So if you eat more than your energy requires, this is where we're going to take these different glucose molecules and we're going to start to store them. If we have excess carbohydrates, proteins, and fats, they're all going to be converted to triglycerides. All of them will, unfortunately. Protein metabolism. Proteins, because a protein is a chain of amino acids. So they're going to get broken down into those individual amino acids. We do not store proteins in our body. These amino acids get oxidized to make ATP or to make new proteins. Excess amino acids get converted into glucose or triglycerides, depending on what our body needs. The two hormones, insulin-like growth factor and insulin, what help or stimulate the transport of amino acids to our cells. So we have proteins in our body that do lots of different things. They can function as enzymes, they can transport, they can be antibodies, clotting chemicals, hormones, contractile elements, and muscle fibers, just to name a few. So we do use a lot of amino acids that we ingest to make a lot of different types of proteins. But a certain amount of protein catabolism is going to happen every day in our body. Cortisol from our adrenal glands are going to stimulate this protein catabolism. So sometimes our proteins just get worn out. They've been used so much that they're no longer effective. At this point, they are broken into amino acids. Some of these amino acids get converted into other amino acids. Hepatocytes convert some of these amino acids into fatty acids, ketone bodies, or glucose. Our cells can then oxidize some of these amino acids to generate ATP. They go into the Krebs cycle at this point. Deamination is where these amino acids get broken into that acetyl-CoA, where they enter the Krebs cycle. So the formation of these different peptide bonds between these amino acids will produce proteins. Amino acids are held together with protein or with peptide bonds. They can be carried on out by ribosomes. Those ribosomes are going to make proteins. This is how we make insulin-like growth factors, thyroid hormone, insulin, estrogens, testosterone. All of these hormones are going to help stimulate protein synthesis, making proteins. Once we have taken in enough dietary proteins, eating more does not increase your bone or muscle mass. Only weight-bearing exercise can increase your bone or muscle mass. This is a common misconception by a lot of people. They think taking in more protein than their body may need will make their muscles bigger. It does not. It gets converted to fats. So there are 20 amino acids in our body. 10 of them are essential, which means we need to take them in. We cannot make them. So complete proteins are specific foods that contain all of the amino acids that our body needs. Beef, fish, poultry, eggs, milk, these are considered complete proteins. They have everything you need in them. Incomplete. They have some amino acids, but not all of them. Like leafy greens, legumes, grains, they only contain a specific amount. And then we have non-essential amino acids, which means these are ones that the body can make through transamination. So all of these different amino acids, this lists all the different types of amino acids, and it can show where these different amino acids can enter into this Krebs cycle to help generate energy. Phenylketonuria. Phenylketonuria is a genetic error in protein metabolism. And if there is elevated levels of this phenylalanine, what happens to these people is their genes are coding wrong for the enzyme that breaks down this amino acid. This amino acid gets broken down into tyrosine. If they do not have this enzyme present, this phenylalanine will build up in the blood. If it does not get treated, this is what causes people to vomit, get rashes, seizures, growth deficiencies, and mental retardation. PKU, phenylketonuria, is now screened for in every newborn. If it is present, if they have this specific genetic error, they need to reduce or restrict your diet so you only get the amount of phenylalanine that is needed for growth. Aspartame, which is found in NutraSweet, aspartame is found in most diet sodas. They contain that phenylalanine. And consumption can be completely restricted, so they cannot have it because of this specific disorder. So there's a lot of different key molecules that play a role in our cell's metabolism. That's that glucose 6-phosphate, the pyruvic acid, and that acetyl coenzyme A or acetyl CoA. These are products that need to be made and converted to enter the next stage in the cycle of our cell's metabolism. So after glucose enters a cell, we have a kinase, an enzyme, that's going to turn it into that glucose 6-phosphate. Glucose 6-phosphate can be turned into glycogen. It can cause the release of glucose into the bloodstream. We can make nucleic acids with it. And we can put it through glycolysis. So there's a lot of different ways we can take these glucose molecules, they get converted to that glucose 6-phosphate, and then they can go to glycogen. They can be released back into the bloodstream. They can make nucleic acids. We can put it through glycolysis and break down to make energy. So that pyruvic acid is that product from glycolysis. We talked about when we looked at glycolysis, what happens. Pyruvic acid is that end product in glycolysis. We have oxygen. Cellular respiration can happen in that cell's mitochondria. If we don't have that oxygen, we make lactic acid. Pyruvic acid gets converted to lactic acid. It diffuses into the bloodstream where the liver can convert it back to pyruvic acid. Cardiac muscle can take that lactic acid and make ATP from it. We can produce alanine. These different carbohydrate and protein metabolisms kind of get linked to this pyruvic acid. So an amino group can be added to that pyruvic acid to produce alanine. It can be removed from alanine to make pyruvic acid. So we can go back and forth. Gluconeogenesis. We can make glucose, pyruvic acid, and some amino acids can be converted to glucose 6-phosphate. They can also be converted to oxalic acid. This oxalic acid can be fed into parts of that Krebs cycle to form glucose 6-phosphate. So that acetyl-CoA. If we do not have an ATP levels are low, but we do but oxygen is plentiful. We have plenty of oxygen. This pyruvic acid is going to go towards ATP production. So it's going to go through that Krebs cycle and that electron transport chain. But first we have to take pyruvic acid and convert it to that acetyl coenzyme A in that reaction cycle we looked at above. The acetyl CoA is what can enter the Krebs cycle. Putting acetyl CoA through that Krebs cycle can give us carbon dioxide, NADH, and FADH2. We can actually make lipids. So acetyl CoA can actually be used to make some of our lipids. Pyruvic acid can get converted to acetyl CoA. Carbohydrates can be turned into triglycerides. And they store those excess carbohydrates as fat. We cannot reconvert acetyl CoA into pyruvic acid. It doesn't go backwards. It's a one-way process. So some fatty acids cannot be used to generate glucose or any other carbohydrate molecule. So the regulation of these different metabolic actions is going to depend on that chemical environment. It's going to depend on the levels of ATP and oxygen that are available. During the absorptive state, this is when nutrients are entering the bloodstream after we have just eaten. Glucose is readily available for ATP production at this point. The post-absorptive state, these nutrients from this digestive canal is complete. We have brought out everything out of what we have eaten that we can. Energy needs need to be met by different fuels, ways our body has to make this energy. It takes about 4 hours to complete the absorption process after each meal. So 3 meals a day, the absorptive state actually occurs for 12 hours a day. If there's no snacking between meals, so those other 12 hours are in the post-absorptive state. So during this absorptive state, some of these different nutrients are used for energy or to make proteins. So the catabolism of glucose, where we break down glucose for energy. About 50% of the glucose that is absorbed from a typical meal will be used to produce that ATP. Catabolism of amino acids. So some of these amino acids are going to go to those hepatocytes. Where they are deaminated into keto acids. Keto acids can enter the Krebs cycle. They can be used to synthesize glucose or fatty acids. And then there's protein synthesis. So a lot of the amino acids that enter the body cells for this synthesis to make our body's own proteins we need. We will catabolize few dietary lipids. So during this absorptive state, only like a very small portion are actually of these lipids are broken down for energy. Most of them are stored in our adipose tissues. These absorbed nutrients in excess are converted to nutrient stores, that glycogen and fat. So the glycogenesis, the building of glycogen, glucose can be stored as glycogen in the skeletal muscles and the liver. Lipolysis the liver can convert glucose or amino acids into fatty acids for the synthesis of triglycerides. So these adipocytes will take up glucose not picked up by the liver and convert them to triglycerides for storage. So about 40% of the glucose is absorbed from a meal is converted to triglycerides. And about 10% of them are stored as glycogen. Those triglycerides get transported from the liver to the adipose tissues. So some of these fatty acids and these triglycerides that are synthesized in the liver, that are made in the liver, remain there. Hepatocytes tend to package most into these VLDLs or LDLs that go to the adipose cells for storage. So when we're absorbing these different nutrients, we can make ATP with some of this glucose. We get stored as glycogen. They can be converted to triglycerides and stored in the fat. So after a meal, glucose-dependent insulinotropic peptide and rising levels of blood glucose levels and certain amino acids actually stimulate that pancreatic beta cells to release insulin. Insulin is used to increase the activity of enzymes and to make different storage materials. So what it's going to do is it decreases the enzymes that are used to break something down, and it promotes making glycogen. It's going to promote the storage of lipids and the synthesis of triglycerides. So glucose enters blood cells through glucose transporters. So high levels of glucose brings more of these glucose transporters to the cell membrane to allow more glucose to enter the cells. During this post-absorptive state, so about 4 hours after your last meal, the absorption of nutrients in the intestines is complete. We're not getting any more nutrients out. At this time, their blood glucose levels start to fall. They start to go down because our cells are using this glucose to make energy, taking it out of the bloodstream to make this different energy. So during this post-absorptive state, the normal blood glucose level could be between 70 and 110 milligrams per 100 milliliters. Our blood glucose level stays within this range through different homeostatic mechanisms. So your nervous system can pretty much primarily uses glucose for energy, because fatty acids can't cross that blood-brain barrier. Red blood cells get all of their ATP from glycolysis of glucose, so breaking down that glucose. They have no mitochondria, so that Krebs cycle and the electron transport chain is not an option for them. These blood glucose concentrations are maintained at that pretty normal level because we can form new glucose from that glycogen, that glycogenolysis in the liver. We store enough glucose in our liver in the form of glycogen. It provides about a 4-hour supply of glucose. Glycogenolysis in the muscles, glycogen, breaking down that glycogen. The glycogen that is catabolized in the skeletal muscles is used to provide ATP specifically for muscle contractions. So that glycogen in the liver, they don't release that glucose outside of, or in the liver, in the muscles, they don't release that glucose out into the bloodstream from the muscles. They use that glucose. Once they get that glucose, it's all theirs. Lipolysis. So we start to break down triglycerides. It gets released out into the bloodstream, and the liver converts it into glucose. That then gets released back out into the bloodstream again. We have our protein catabolism. So we can break down proteins in skeletal muscle and other tissues to release out amino acids. These amino acids can be converted to glucose in the liver, and then gluconeogenesis, forming new glucose molecules from non-carbohydrate sources. Lactic acid, glycerol, amino acids. This is the formation of new glucose. These are all different things that we use to get ATP during this non-absorptive state to maintain our blood glucose levels during this non-absorptive state. So glucose sparing. So this is happening when most of our cells switch to some other fuel. They use something else other than glucose for energy. So it leaves more glucose for the brain and the red blood cells because that's all that they can utilize. So that's where they start using fatty acids. So that lipolysis, breaking down of lipids, it cannot be used for glucose production because that acetyl-CoA cannot be turned into pyruvic acid very readily. So most of our cells, they can catabolize these fatty acids and they feed them directly into that Krebs cycle. Catabolism of lactic acid. Cardiac muscle, as I've been saying, the heart can produce ATP from lactic acid. That catabolism of amino acids. So those liver cells, those hepatocytes, they can break down those amino acids to produce ATP. Catabolism of ketone bodies. Again, our hepatocytes and our liver convert fatty acids to ketone bodies, which then gets released out into the bloodstream, and the heart, the kidneys, and other tissues can use those ketone bodies to make ATP. So there's a lot of different ways that we can maintain our blood glucose levels. There's a lot of different ways that we can produce energy. Our brain and our red blood cells can only utilize glucose. So our other cells sometimes have to use other ways to get energy to spare that glucose for your brain and your skeletal muscle or your red blood cells. So there's different hormones that are going to regulate your metabolism when we're not absorbing these nutrients. That sympathetic division of your autonomic nervous system is also going to help regulate that. So anti-insulin hormones can counter the effect of insulin. So as your blood glucose levels go down, the secretion of insulin falls, which causes the release of this anti-insulin hormone to rise. So when this blood glucose concentration starts to decline, pancreatic alpha cells release that glucagon. That glucagon is going to go to the liver, and it's going to increase the release of glucose into the blood. Low glucose will also activate that sympathetic system. So, because our neurons need glucose for energy. If these glucose sensitive neurons detect low blood glucose, it'll increase that sympathetic output. It raises the epinephrine and those norepinephrine levels, which causes glycogen breakdown, which will also start to stimulate that lipolysis as well. In stressful situations on your body, when we have low blood glucose, that is a stressful event on your body. Hot or cold temperatures, fever, trauma will actually cause cortisol to be released. Cortisol promotes gluconeogenesis, lipolysis, and protein catabolism as ways to get our energy for our cells. What about people who fast? Fasting is going without food for many hours or days. Starvation is weeks to months of food deprivation or inadequate food intake. So you're taking in food, but you're taking in candy. You're not taking in the proteins and the other nutrients that your body needs. That's technically starving your body. You're just giving it lots of sugar if you're taking in candy, as an example. People can survive for about 2 months without food, as long as they drink enough water. Glycogen stores tend to be depleted within a few hours when you're fasting. So we start breaking down triglycerides. We start breaking down structural proteins for energy after several weeks. Then we start breaking down organs and muscles. The amount of adipose tissue will actually determine the lifespan possible without food. So during this starvation or fasting, this nervous tissue and these red blood cells, they're able to still use glucose for energy. But by the second day you're fasting, at this point, your blood glucose levels start to stabilize out. We have more fatty acids that are found in our blood at this point. We start to form ketone bodies. And so by day 40 of starvation, ketones provide most of the brain's energy. So how do we balance this energy expenditure? So when energy content in food balances the energy used by all cells, the body's weight remains constant. So you don't gain weight, you don't lose weight, as long as you're giving your body the amount of energy it needs every day to get by. In many people, weight stability persists despite very large day-to-day variations. They can eat a lot one day, a little bit the next. But in most affluent nations, a very large fraction of the population is overweight. Being overweight increases the risk from dying from a variety of cardiovascular and metabolic disorders, hypertension, varicose veins, diabetes, arthritis, cancers. These are more common in the overweight population. So calories. Calories are the amount of energy in the form of heat required to raise the temperature of 1 gram of water 1 degree Celsius. That is technically what a calorie is. It's either known as a kilocalorie or a calorie with a capital C will be used. And these are kilocalorie or calories are often express the energy contents of food, which is in the form of about 1,000 calories. So we say food contains 500 calories. We're technically referring to kilocalories. So all these different kilocalories in our food come from carbohydrates, proteins, and fats, breaking these down. The catabolism of proteins and carbohydrates give approximately 4 kilocalories per gram. Fats are about 9. Alcohol is about 7. So the number of kilocalories can be calculated by multiplying the number of grams of that component by its energy content. So the higher the calorie content, the greater amount of energy being released. Then comes in our metabolic rate. This plays a role into how many calories your body can use. your metabolic rate is all of these different reactions that use energy. Your hormones, like your thyroid hormone, is going to be a big player in this because your thyroid hormone actually sets your basal metabolic rate, that how many calories your body burns at rest. This basal metabolic rate increases the blood levels of thyroid hormone. So the response to this change in levels of this thyroid hormone, it's very slow. It takes time. So thyroid increases that basal metabolic rate. And it does this by stimulating cellular respiration. So how much ATP your cells use, and it raises your body temperature, which is the chlorogenic effect. Exercise. So strenuous exercise, your basal metabolic rate can increase by up to 15 times that basal rate. In a well-trained athlete, theirs may raise up to 20 times. Your nervous system. So during exercise, or even some stressful event, that sympathetic division stimulates the release of that norepinephrine and that epinephrine, and those actually increase your cell's metabolic rate as well. Body temperature. The higher your body temperature, the higher your metabolic rate. For every 1 degree Celsius your core temperature raises up, it increases those chemical reactions by 10%. So that's a pretty large jump. The ingestion of food is going to change your basal metabolic rate. So as we take in food, it can raise your metabolic rate 10 to 20% because the energy used to digest, absorb, and store these nutrients. This is food-induced thermogenesis. Your age. The younger you are, the higher your metabolic rate. The metabolic rate of a child is about double that of an elderly adult. Gender. Females tend to be lower except for pregnancy and lactation. Your climate. Do you live in a tropical region? Do you live in the Arctic? Your sleep. How much sleep you're getting. Malnutrition actually lowers your metabolic rate. So there's a lot of factors that are going to affect it. So it gets really hard to calculate out this basal metabolic rate because there's so many different factors. That is this basal state. So this basal metabolic rate is measured through these different conditions. So the most common way, by measuring the amount of oxygen used per kilocalorie of food metabolized. So when we use 1 liter of oxygen to catabolize different dietary mixtures of triglycerides, carbohydrates, proteins, the basal metabolic rate for an adult is typically between 1,200 to 1,800 kilocalories per day, or 24 kilocalories per kilogram of body mass in males and 22 kilocalories per kilogram in adult females. So since we don't use kilograms, we'd have to convert our weight over to kilograms first before we are able to figure this out. So then the added calories needed to support daily activities, digestion, walking, it can be anywhere from 500 kilocalories for a sedentary person to over 3,000 kilocalories for someone training for an Olympic level competition or mountain climbing, because mountain climbing takes quite a bit of energy. If you've never tried it, it's fun. It does require a lot of energy though. So that total metabolic rate, that total amount of energy you use by the body per unit of time. So again, we use that basal metabolic rate, and that's about 60% of that total metabolic rate. Whatever physical activity, that's about another 30 to 35. Lower if you're more sedentary. Non-exercise activity thermogenesis. So the energy costs to maintain that muscle tone, your posture, standing or sitting. Involuntary fidgeting movements. You a high fidgeter? That can help you burn more calories. Food-induced thermogenesis. So that's the heat produced while this food is being digested, absorbed, and stored. So here's a list of a bunch of different activities and generally how many calories are expended per hour. It's different for every person, of course. So this is just kind of a general guide average. Canoeing, aerobics, reading, playing the piano, housework, office work, cleaning, running versus walking, sitting versus standing, studying, swimming. Swimming consumes quite a bit because there's a lot of resistance to it. Texting even. You can burn 40 calories an hour. You're texting for an entire hour. So we store most of our chemical energy in our fat cells, those adipose cells. So when energy use exceeds energy input, triglycerides get broken down for energy. So when energy input exceeds expenditure, any of that excess energy is stored as triglycerides. So the amount of triglycerides that are kind of indicate how much extra food you're taking in. That small amount, you may think it's a small amount, but it adds up. So a gain of 20 pounds between the ages of 22 to 55 usually only shows a very tiny imbalance over time. So we use a lot of different negative feedback mechanisms that are going to help to regulate our energy intake versus our energy expenditure. There are no sensory receptors that are going to monitor our weight, our body size. This is all done through feeling full. Hopefully, you feel that sensation when you are eating. So we have some neural and endocrine signals that try to help regulate food intake. So these different nutrients that are found in the blood, plus different physical elements, your stress levels or depression. These are going to send signals from this digestive canal, and then your special senses, what you're seeing, hearing, smelling. This is all going to go to the hypothalamus and to other areas in the brain to try to help with this satiety, which is feeling full, which should cause a lack of desire to eat. The arcuate nucleus and the paraventricular nucleus and the hypothalamus are supposed to help regulate food intake. Leptin. It's supposed to decrease adiposity or decrease total body fat mass. Our fat cells, those adipose cells, actually make this leptin, and it goes to the hypothalamus to inhibit the circuits that are going to stimulate you to eat. These two feelings, feeling of full and this leptin hormone are supposed to help to regulate your food intake. So when leptin and insulin levels are low, these neurons from the arcuate nucleus and the paraventricular nucleus are going to release neuropeptide Y. This stimulates you to eat. Melanocortin, another neurotransmitter, these actually are released, and it's pretty similar to melanocyte stimulating hormone. We have ghrelin. This is produced by the stomach, and this works to increase your appetite. So this energy balance actually requires the regulation of energy intake, taking in those calories. So most increases and decreases in food intake are usually due to the different sizes and meals instead of changing the number of meals. There's a lot of different experiments out there that are going to demonstrate the presence of satiety signals, feeling that full, chemical and neural changes, and then termination of eating when you're feeling full. Does that happen? Here's the problem. A lot of people stress eat. A lot of people emotionally eat. Socially eat. You're around social events and there's food out on the table. It causes you to eat. We celebrate, punish, comfort, defy, and deny ourselves food, which can lead to emotional eating. It's very common. Almost everybody does it in some form or fashion. That's why it's considered within the normal range of normal behavior. So when you're emotionally eating, when it becomes excessive, this is when it interferes with your health. This is when it causes obesity. It can cause hypertension as these fat and cholesterol kind of get deposited around these blood cells or in the blood vessels, heart disease. It can be associated with poor self-esteem, the inability to cope with stress, even eating disorders like anorexia or bulimia. For a lot of people, eating provides comfort. It can numb the pain, any kind of pain you're going through. So people who emotionally eat tend to overeat carbohydrates because when you really eat carbohydrates, it raises the levels of serotonin in your brain and it leads you to feel good, feel happy, feel relaxed. So food starts to become a way for a lot of people to self-medicate. This is where most people tend to struggle with taking in enough calories for your body to survive versus eating more. Your body produces most of the heat depending on these metabolic reactions. A lot of these metabolic reactions are going to release out heat, and the release of this heat regulates your body temperature. So that homeostasis of this body temperature can only be maintained as long as the rate of heat loss from the body equals the rate of heat production. So heat, it is a form of energy, and it's measured as temperature. So when you take your body's temperature, we're actually, besides checking to see if you have a fever, you can actually check that heat through these different chemical reactions. So your core temperature is that temperature your body sits at. It's that subcutaneous tissue. It's that temperature that sits in there. That shell temperature is the temperature near your body's surface. So that's skin, that's subcutaneous tissue, and it is usually 1 to 6 degrees Celsius lower than the core temperature. So if your core temperature gets too high, it can kill proteins. If it's too low, it can cause cardiac arrhythmias that also result in death. So to maintain our body's temperature, it's going to kind of depend on the ability to lose heat to the environment at the same rate we can produce it. Conduction is that heat exchange that's going to occur between molecules of two materials that are in direct contact with each other. So when we're at rest, about 3% of your body heat is lost in conduction to cooler material. So you're resting your hand on a table or a desk, it's probably cooler than your body temperature. So some of the heat from your body temperature is transferring over to this inanimate object, that table. We can also gain heat this way. You're outside on a hot blacktop, heat gets absorbed up into your system. Water conducts heat a lot more effectively than air does. Convection. This is the transfer of heat by the movement of air or water between areas of different temperatures. So the contact of air or water with your body results in the heat transfer by conduction and convection. When cool air hits you, you're outside on a hot day, but there's a nice cool breeze, it actually warms that air when it brushes across your body. Radiation. Radiation is that transfer of heat in the form of infrared rays. So some things become warmer through radiation. So your body will lose heat by radiating more of these infrared rays than it's going to absorb from other objects. So if there's objects surrounding you in somewhere that are warmer than you, you will absorb more heat than you lose. There's also that evaporation, that conversion of a liquid to a vapor. This is our insensible water loss that we lose through that insensible perspiration. We will lose it through the mouth, through the respiratory system. We're unaware that we're losing this water. So if you're in a place that has a higher humidity, the evaporation rate is lower. When you're in 100% humidity, we gain heat via condensation of water. So we sweat it, or water can collect onto your skin. We gain heat that way. And this is how we prevent overheating during exercise, is during this evaporation of water off of our skin surface. Our hypothalamus is our body's thermostat. Your hypothalamus is going to regulate your body's temperature. It is in the anterior part of the hypothalamus by that preoptic area. So it's going to receive input from different thermoreceptors. So then it's going to send out information. So when blood temperature increases and lower frequencies when blood temperature decreases. So these nerve impulses are going to go to the heat losing center or the heat promoting center within this hypothalamus that regulates this thermostat. And it's going to set different responses that are going to change. They're going to lower or raise your your temperature based off of these different signals that are being sent. So if your core temperature declines, your body temperature goes down too far. We do have different ways to try to conserve heat. to keep some warmth within your body. And again, it is a negative feedback that is causing this to happen. So we have nerves that come from that hypothalamus, and this thyroid stimulating hormone that gets sent out to different cells, and it's going to work to vasoconstrict blood vessels. We're going to take that blood that is going to the skin and transfer it to the internal body, to your core organs, to your vital organs, to keep them functioning well. We release epinephrine and norepinephrine. This brings an increase in cellular metabolism. The cells are increase in cell metabolism increases the amount of heat that is produced. Then shivering. Every time you contract a skeletal muscle, it generates heat. So maximum shivering can raise your body temperature about 4 times the basal metabolic rate in minutes. And then that thyroid hormone, that thyroid stimulating hormone mentioned, it goes to trigger the release of thyroid hormone, which is going to increase the metabolic rate, which will increase your body temperature. So if the core body temperature starts to rise above normal, again, negative feedback loops to bring it back down. So the higher the body temperature, this blood is going to stimulate peripheral and central thermoreceptors. Again, these thermoreceptors are going into that preoptic area where that body's thermostat, and it starts to stimulate stimulate the heat losing center. So it's going to dilate the blood vessels closer to your skin surface. And this causes excess heat to be lost out through radiation and conduction. So this whole process is going to work through this negative feedback to cause you to either warm up the body temperature or cool it back off, depending on what your body needs at that time. Hypothermia. Hypothermia is when your core temperature it falls to 95 degrees Fahrenheit or below. It could be cold stress, like immersion into cold water, metabolic diseases like hypoglycemia, hypothyroidism, drugs like alcohol, antidepressants, sedatives, tranquilizers, burns, and malnutrition. So when your core body temperature falls below 95, you feel cold, you shiver, you can start being confused. Your vasoconstriction, your blood vessels constrict so the blood stays to your core organs, your muscles get stiff. Your heart rate slows down. It can cause that acidosis, hypoventilation, hypotension, low blood pressure, low breathing rate. You lose any spontaneous movement. It can lead to coma and eventually can lead to death. So nutrition. These are all the different chemical substances of food that our body needs for growth, maintenance, and repair of all of our cells. We have water, carbohydrates, lipids, proteins, minerals, and vitamins. These are the different types of nutrients our body needs. We need 2 to 3 liters of water a day because we're losing water all day long. Lipids, carbohydrates, proteins, these give our cells energy. So those essential nutrients are the nutrients that our body cannot make, so we have to take them in. That includes some amino acids, some fatty acids, some of those vitamins and minerals. They're essential nutrients. So for each gram of protein or carbohydrate, that gives us about 4 calories. 1 gram of fats gives us 9 calories. But we don't know with certainty which types of carbohydrates, fats, or proteins are optimal in the diet because everybody is so different with what their body needs. Different populations around the world eat such different diets that are adequate to their lifestyle, that it's really hard to make these types of recommendations for that as well. So people, they say about 50 to 60% of carbohydrates with less than 15% from simple sugars. We need less than 30% of fats with no more than 10% saturated. And 12 to 15% of our total diet should be protein. In 2011, they went from that food pyramid recommendation, they switched over to what is now called My Plate. And in this My Plate, they kind of physically show how much of your plate should be covered in fruits, vegetables, grains, proteins, and dairy. This was done to give more of a visual of an actual plate and what it should look like versus that food pyramid. It's a lot harder for some people to understand how much of their plate should be carbohydrates versus proteins versus fruits. So for this specific reason, they switched over to this type of diagram instead. So among these different guidelines, they say to enjoy food, but balance your calories and eat less. Avoid oversized portions. Try to make half your plate fruits and vegetables. Switch to fat-free or low-fat milk. Make at least half of your grains whole grains. Choose foods with lower sodium. And drink water instead of sugary drinks. Why they turned it into that picture of the plate is because they're trying to get people to eat proportions. In moderation, eat variety, and try to eat some nutrient-dense foods. This was their main reason for switching to this visual versus that food pyramid. So minerals. These are inorganic elements that naturally occur at the Earth's crust. So in our bodies, these different minerals seem to appear in combinations with other minerals. And they make up about 4% of your total body mass. We see them in the skeleton in the form of calcium and phosphate, potassium, sulfur, sodium, chloride, magnesium, iron, iodide, manganese, copper, cobalt, zinc, fluoride, selenium, and chromium. These are the minerals that help your body function in day-to-day life. Your body can uses these different ions rather than non-ionized forms. So they use these in more in an ionized forms. So your diet should have an adequate amount of potassium, sodium, chloride, and magnesium. Minerals don't form these long chain compounds, so they're not really good building materials, but they help with enzymatic reactions. Like magnesium, it's a catalyst for ADP to ATP. So it is used to help form and speed up these ADP to ATP reactions. Vitamins. Vitamins are other types of nutrients that are required for growth and metabolism. Vitamins don't provide you with energy. They do not serve as building blocks to your body. They usually are some sort of coenzyme. When a vitamin attaches to an enzyme, it causes that reaction that enzyme needs to do to actually happen. Our body can't make most of them. Vitamin K we do get from the bacteria in our digestive tract. We can assemble some of these vitamins. Those are known as provitamins. Like vitamin A gets produced from beta carotene. There is no single food that is going to contain every single vitamin that our body needs. Because we have fat-soluble vitamins like A, D, E, and K. They cannot be absorbed unless they are actually ingested with lipids. Water-soluble vitamins, like most of our B vitamins, vitamin C, these do dissolve in body fluids. We cannot store the extra ones, so the vitamins that we don't use get excreted in the urine. I had a friend who used to buy these very expensive vitamins, and she noticed her urine color used to get very bright. She realized that these expensive vitamins were making very expensive urine for her, and decided that she needed to take vitamins in different ways because the way these vitamins work did not work for her. So we do have vitamins that act as an antioxidant. They help to get rid of or inactivate oxygen-free radicals, like superoxide. C, E, and beta carotene. These vitamins, these free radical damage in the cell membranes, are very reactive. So these antioxidant vitamins help to protect against some form of cancers from these free radicals. They reduce the buildup of atherosclerotic plaques in your blood vessels. They can delay some of the effects of aging. And they can decrease the change of cataract formation in the lens. So vitamins are very beneficial to your body, and they are very needed. So these charts here go through some of the things that can happen with a deficiency or too much in your system for fat-soluble vitamins. Some of these are good to read. Like a deficiency in vitamin A can cause night blindness, where you're not able to see well at night. A deficiency of vitamin D can cause rickets or osteomalacia, which is decreased bone density, and you can actually lose muscle tone from this. Vitamin E from hemolytic anemia. K is used in clotting factors, so it can cause excess bleeding. Then our water-soluble vitamins. There's a lot of different things that these different water-soluble vitamins, where they come from, what they can lead, like what they do and what they lead to if you don't have enough. Niacin is a type of B vitamin. B12 can lead to pernicious anemia if you don't have enough. It can lead to memory loss. It can impair your osteoblasts so they don't work well. Pantothenic acid can cause fatigue and muscle spasms. It can cause insomnia. So these water-soluble vitamins get eliminated in the urine if you take too much. So vitamin C, some of those B vitamins. Biotin is another B vitamin. Can cause depression if you don't have enough. It can cause that fatigue and muscle pain. So vitamins and mineral supplements, we try to tell people to try to get as many of their vitamins and minerals as they can through natural sources, so through foods, rather than taking supplements. It does happen that people do need to take vitamins, and sometimes it has to do with enzyme deficiencies, that they need vitamins taken into their system in specific forms for their body to be able to use it. Hypervitaminosis. This is if you get too much of specific vitamins, and it actually exceeds the ability of the body to utilize, store, or excrete. This usually happens with lipid-soluble vitamins. They do get stored. So if you take too much, it can actually cause problems. Like too much vitamin A can cause drowsiness, weakness, irritability, headache, vomiting, peeling skin, hair loss, joint pain. There's a lot of different things that excess vitamins can cause. Hypervitaminosis, hypovitaminosis. All right, that is a deficiency of different vitamins. You can check most of these through different blood tests, especially these fat-soluble vitamins. A lot of these water-soluble vitamins too, you can actually check these through different blood tests. So anorexia nervosa. This is actually a disorder with self-induced weight loss. These people have a very negative body image. And they end up having a lot of nutritional depletion. They kind of have this fixation on their weight, that they constantly eat too much. Well, they really don't eat too much, that they have too much body fat on them, even though they may really have none or next to none. They insist on having bowel movements every day, even though they're not taking in food to form bowel movements. They can abuse laxatives, which cause fluid imbalances. It's more common in young single females, but it can be inherited. They can have abnormal menstrual cycles. They can even have amenorrhea, which is a complete absence of a menstrual cycle. They usually have a lower basal metabolic rate because they're not eating. If it's not treated, they will die of starvation or one of the complications to it. They usually end up with osteoporosis because they're not getting enough calcium and phosphorus in their diet. They're not getting enough hormones being released to maintain their bone density. They're usually depressed. They could have some brain abnormalities. Spend a lot of time in psychotherapy and diet regulation to understand that they need to have muscle on their body. They need to have some fat on their body to protect themselves, protect their organs. It takes a lot of retraining of their brain that it's okay to have a normal size body. A fever. We mentioned fever in the lymphatic system. A fever is technically a resetting of that hypothalamic thermostat. Viruses and bacterial infections or bacterial toxins tend to cause it. Ovulation. During ovulation, it can actually cause the body temperature to elevate. Too much thyroid hormone. Tumors. Sometimes people, when they get vaccines, having a fever can be a side effect. So these phagocytes, when they take in bacteria, they secrete a pyrogen. A pyrogen will go into that hypothalamus, and then it'll help to reset the body temperature to higher. Antipyretics like aspirin, acetaminophen, ibuprofen are used to reduce fevers because it inhibits different prostaglandins that are going to cause that hypothalamus to reset its core temperature. If the core temperature rises up to 44 to 46 degrees Celsius, it can actually result in death. But up to a point, fevers are beneficial because they're going to intensify those interferons, those phagocytic activities of macrophages. It increases the heart rate. Infection-fighting white blood cells get delivered to the areas faster. Antibody production and T cell proliferation increases. It's going to speed up the rate of chemical reactions. Can help the body repair itself much faster. So fevers are beneficial to a point. Obesity. Obesity is having more than 20% above a desirable standard due to excess accumulation of adipose tissue. More than 1/3 of the US adult population is obese. That's a very large number. Moderate obesity is very hazardous to yourself. It can cause cardiovascular disease, hypertension, that increased blood pressure, pulmonary disease, non-insulin dependent diabetes, so that type 2 diabetes, arthritis, that extra weight putting down on especially the knees. That's usually the first place you see arthritis in obese people is in their knees because of all that extra weight their knees are carrying. Breast, uterus, colon cancers get increased, varicose veins, gallbladder disease. Sometimes obesity can result from trauma or tumors in that hypothalamus. There's that food regulating center. But most of the time, there's no specific cause. Genetics can play a role if your parents are obese. Eating habits that are taught to you through your parents. Stress eating. Social customs. Obese people tend to burn fewer calories during that digestion and absorption of a meal. So a smaller food-induced thermogenesis effect. They don't burn as much. So these obese people who do lose weight, they require 15% fewer calories to maintain a normal body weight than people who have never been obese. People who gain weight easily, they exhibit less non-exercise activity thermogenesis. So leptin is not deficient in most obese people. Most of a surplus, too many calories that get converted to triglycerides and stored in those adipose cells. Those adipocytes increase in size. When they reach their maximum size, they start to divide and create more adipose cells. So as these adipocytes proliferate, continue to divide, this can occur in extreme obesity. Lipase, an enzyme for fats in the that's active in the abdomen, but it's less active in hip fat. So this accumulation of fat in the abdomen brings on higher blood cholesterol levels. It brings on other cardiac risks. Treatment is difficult because most people who successfully lose this weight tend to gain it back within 2 years. Once you've been obese, it's very easy to get back there. So even a moderate weight loss is associated with different health benefits. It takes a lot of behavior modification programs of eating, monitoring, and recording a very low calorie diet. There's different drugs, and then those different bariatric surgeries that we talked about in the digestive system as well. Thank you very much for listening. Have a wonderful rest of your day. Thank you. Bye.